Hi everyone, this is Miss Lasar, and today I'm going to be teaching you about sampling methods. This set of notes is set up in outline format, which you're familiar with. Make sure your numbers and letters are vertically aligned. Pay attention to how much you've tabbed in, keep it consistent for each level. If it's in black, it's text that you are required to write down. And if it's in blue, it's optional to write down. Maybe it's a fun fact, maybe it's an example. You choose whether you write it down. Remember to pause the video to write down notes when you need to, because I will definitely be going too fast for you to do this if you're just listening to me. Here we go. First section capital A, what is sampling? Well, we need to define two things before we can define sampling. First, a population. Detail number one, population. This is a vocabulary word. I've bolded it. You should underline it or highlight it, make it stand out. A population is all members in a group of the same species. A sample is a subset of members in a specified population. So here's our population. We've taken out just a few. That's our sample. So detail 3, notice this is still within section A. Make sure this 3 is vertically aligned with the 1 and 2. Sampling is taking a subset of a population and making conclusions about the population as a whole. For example, when we track animals and we put tracking tags on just a few migratory birds and watch the patterns of how they migrate, we're inferring that their migration is pretty similar to the migration of the rest of the flock that we didn't tag. And so we can make inferences about the whole population of migratory birds by just looking at the data from a few. When we develop vaccines or medication, we take a sample of people and we put them in a clinical trial and we give them the vaccine or the medication. We infer that their reaction to the drug or vaccine will be similar to everyone else's. And so how they respond to the vaccine or medication will be the same as how the rest of the population responds to the vaccine or medication. Section B, why do we sample? Well, two simple reasons. Number one, we can't count the whole population, or if we could, we can't do it fast enough. Detail 2, we need information fast. So here are 3 real types of questions that you would need to use sampling to answer. Our first question, how many wildebeest in the world have rinderpest right now? Rinderpest is a deadly virus. We need to use sampling to do this because we can't count the whole population of wildebeest, and we need this information quickly so we know how to respond. What percentage of people in Maryland have already been infected with COVID-19? Can't look at every single person in Maryland, so we need to use sampling to make an estimation for this number. How many invasive lionfish are in the Caribbean? It's too big for us to survey all of it. So we'll use sampling to make an estimation. Here's a slightly more in-depth example. Back to our question, how many wildebeest in the world have rinderpest right now? Our population is all the wildebeest in the world. Is it possible to count and diagnose all of them with rinderpest? No, that would be too labor intensive, too costly, take too much time. We maybe would even have some of the wildebeest die or be born while we're in the process of doing that. No, we can't do this. So we need to sample. Sampling will look something like this, and I'll give you more specific techniques later in this video. We'll take a fixed number of wildebeest, that's our sample. We'll determine how many in the sample have rinderpest, and we'll use that to infer, to estimate a positivity rate for the rest of the population. And we'll talk about more of the details shortly. What can we measure with sampling? This is section C, capital C. Make sure it's vertically aligned with the capital A and capital B. We can measure and determine 3 big things. Number 1, population size. We can use sampling to infer the population size of a particular population, and I'll show you how. Number 2, we can use it to infer population density, how closely packed the population is. And number 3, we can use it to infer the percentage or the number affected by something, like what percentage of the population of wildebeest have rinderpest. Population density is one that comes with a formula. Go ahead and write down this formula to the right of where you wrote down population density. Population density equals the total number of individuals divided by the area that they're in. And we're going to use this formula momentarily. Just put a box around it, make it stand out in your notes so that you can easily find this formula again later. Here are some examples of population calculations. This is in blue. The question text will be in blue. The actual solution will be in black. So you are required to write down the solution. It is optional to write down the question text. If it helps you remember what you did, feel free to write it down, but know that this is optional. What is the population density of trees in a 50-acre forest if 80 trees are counted in 2 acres? This is a population density calculation. Here's our formula. Total number of individuals divided by the area that they're in. Well, there are 80 trees in 2 acres. So 80 trees divided by 2 acres is 40 trees per acre. And that's our answer. Next question. What is the total population size of the trees in a 50-acre forest if 80 trees are counted in 2 acres? Okay, so we just calculated 80 trees in 2 acres is what we have. Well, that's the population size for 2 acres. We want it for 50 acres, so we'll multiply by 50 acres. Acres will cancel out and we'll be left with just the final unit of trees, which is what we want for population size. 80 times 50 divided by 2 is 2,000 trees. That's our estimate of the total population size. Here's a more complicated question that we don't have the tools to answer yet, but you will. How does the population density of trees change as one moves away from the road? As our habitat changes, how does our population density change? And you'll get some tools to answer this momentarily. We're ready for our next section. This is our longest section, section D, types of population sampling. There are 4 main types. For each type, you're going to write down the method, what it's good for, and what it's bad for. Here's our first type of sampling, quadrat sampling. And a quadrat is actually this device right here. It's a device in the shape of a square or a circle that's got a fixed size and is empty in the middle. So we can put it down and we can see what's inside of it, just like our diver is doing in this image, and just like our ecologist is doing off screen in this image. Here's how it works. Section small a is method. Detail 1, we're going to define a sampling region. And I'll show you a whole example of this in a moment. For now, just write down these steps and it's okay if you're a little confused. Number 2, after we've defined a sampling region, select random areas that are the size of 1 quadrat. And we're going to do this by usually throwing our quadrat and having it land in a random location. Number 3, count the individuals inside each quadrat. And number 4, use this to calculate our population density or our population size. Remember, when I say quadrat, and I'm going to say it a lot, the quadrat is this device that is a square or a circle of fixed size. You could even use a hula hoop for this. And it's empty in the middle. So we're going to put it down on the ground or on the bottom of the ocean. And we're going to count what's inside this. Here's our example. Whoa, this is a packed field with lots of stuff. I see broadleaf plantain, clover, and ground ivy. What if I wanted to determine the population density of each of these? Well, here's a table that I'm going to calculate this in. This is all in blue, so it's optional to write down, but this might be helpful to have in your notebook for later. What I'm going to do is I'm going to throw my quadrat down somewhere random on this field. There it is. And my quadrat is labeled 1 meter square because I happen to know that its area is 1 meter square. And I'm going to use that area later. And now I'm going to count what's in it. I'm going to count things that are mostly inside the square. I count 1, 2, 3 ground ivy. Record that. I counted 7 clover and 2 broadleaf plantain. Now I'm going to repeat that. I'm going to take that quadrat. I'm going to throw it somewhere else random. Count what's in it. I see 2 ground ivy, 13 clover, 1 broadleaf plantain. Do it again. Here it is. 3 ground ivy, 9 clover, 3 broadleaf plantain. Do it again. New location. 3 ground ivy, 12 clover. It's packed. And 1 broadleaf plantain. And one last time, I see 3 ground ivy, 6 clover, 1 broadleaf plantain. Now I'm going to calculate my averages. 3 plus 2 plus 3 plus 3 plus 3, all divided by 5 is an average of 2.8 ground ivy. My clover is an average of 9.4, and my broadleaf plantain is an average of 1.6. So these averages are what I can use. Here's how I would write this up. There is an average population density of ground ivy of 2.8 plants per meter squared because my area is meter squared. I had an average of 2.8 plants in my 1 meter square quadrat. Now, what if I wanted to calculate population size? Now, just like in the earlier example, I need to know the total area that I'm looking in. And my area, it's a 5 meter by 3.5 meter plot. So my area is 3.5 meters times 5 meters, 17.4 meters squared. And now I can multiply my population density by my area. Meters squared cancel out, and I have 2.8 times 17.4, which gives me approximately 49 plants. My population size of ground ivy in this area is about 49 plants. So returning to your required notes, we're still in quadrat sampling. Now in section little b, what is it good for? Well, it's good for non-moving populations because if we put down our quadrat, they're not going to hop out. It's good for evenly distributed populations because, so this picture actually shows a non-evenly distributed population. What if we put down our quadrat in one of these empty areas of rock and we thought, oh, there's no mussels here. See, no mussels live here. And then we put down our quadrat here and we're like, the mussels are covering the ground. This is not a good population to sample with quadrat sampling because it's so clumped and not evenly distributed. We might be led a little astray. It's good for determining population size, like you just saw, and for determining population density, like you just saw. What is it bad for? Moving populations. If we put down our quadrat and our organism runs out, hops out, or just moves to another area where we might count them again later, no good. It's also bad for clumped or non-randomly distributed populations like we saw here. Here's a more evenly distributed population of wildflowers that would be really good for quadrat sampling. And finally, it's bad for large survey areas. When I say large, I mean the entire state of Wyoming. You need to be able to physically go there and see the whole survey area in order to use quadrat sampling for it. Here's our second type of sampling, transect sampling. In transect sampling, detail A, here's our method. 1, we stretch a line across a sampling site. Notice this tape measure that I have stretched across the ground here. Number 2, I count organisms at even intervals along the line. Notice there's actually quadrats at even intervals along this transect line. Number 3, I repeat that along new lines that are parallel to the first. So I just take this whole operation, move it a couple feet to the right, repeat and count in each quadrat. Move it a couple feet to the right, repeat and count in each quadrat. This method is good for situations where we think the population is going to be changing along the line, and I'll have you write that down momentarily. Here's another picture that shows what transect sampling looks like. We stretch down a line, we walk along that line, and we record what we see as we walk along that line. Then we move our line to a new parallel location, and we repeat the process. Stretch out a line, walk along the line, recording what we see as we walk along the line. Transect sampling, small b, is good for non-moving populations. If it moves to a new line, and we'll count it again later, that's no good. Populations that are distributed in a gradient or pattern. Here's transect sampling in a rocky intertidal zone. So this is an area that's sometimes covered by the waves, sometimes uncovered. We think that the populations will change as we move further away from the ocean. And so we're going to stretch out our line and see, well, how does the population change as we move further away from the ocean? It's also good for determining a rough survey of the biodiversity in an area by walking along the line and counting what we see. Transect sampling is bad for moving populations for the same reason as quadrat sampling. Very large survey areas, we need to actually put down a line. We can't put down a line across the state of Maryland. And 3, determining population size and density. Transect sampling is really much better for seeing how populations change or determining a very rough estimate of biodiversity. Here's our third type of sampling, and the text is smaller because this one has our longest method. Detail 3, sampling type 3, mark and recapture sampling. Here's the method. Number 1, we're going to trap individuals. For example, we're going to go to a pond and go fishing. The fish we catch are the fish that are trapped. Number 2, we're going to count the individuals that we caught. How many fish did we catch? Number three, we're going to mark each individual with a tag or some other marking, and then release all the individuals back into the wild. So we take all those fish that we caught, we put little tags on them, and we release them back into the pond. Number four, we're going to trap the individuals again. So a couple days later, everybody's all mixed up again. We go fishing again and we see who we catch. Number five, we're going to count our total individuals. So, how many did we catch in this second go? And how many have already been marked? In our new sample, you can see some have tags and some don't have tags. What's our total? And how many already have tags on them? Then we'll release all the individuals back into the wild and we'll use those numbers to calculate the population size. There's a formula for this. Let me show it to you now. Oh, before I show you the formula, I forgot. For trapping, we're going to use different trapping methods based on the animal. For fish, we might just go out and catch them. For things like amphibians and salamanders, we might use pitfall traps that they stumble into and then get caught in a deep container. Or we might use aquatic traps for something that swims in a river or stream. They swim into this box and then they can't escape. Here's a trap that we're using to trap sea lamprey, which are invasive and live in rivers. And for tags, we use different things. Here's a radio transmitting tag that we might put on the fin of a fish. We might put an ear tag onto a bear. That ear tag, just like a pierced ear, is not going to hurt them. We might put a little tag onto the skin of an amphibian, or we might actually cut off one of their toes as an alternative if the tag won't stick. Or we might put a sticky tag onto the delicate wing of a butterfly. We can't punch any holes into this, it will die. So, like I said, there's a formula to calculate population size for mark and recapture. Here's the formula. Write it down, put a box around it so it stands out. In mark and recapture, the total population is equal to the number in our first capture times the number in our second capture, divided by the number of marked animals in the recapture, the number of marked animals that are recaptured. In this example, we initially caught 10 fish and we marked them all. In our second capture, we got 10 more fish, but only 5 of them had already been marked. So those are our 3 numbers. Let's plug those in. Number in the first capture is 10, number in the second capture is also 10, and the number of marked animals recaptured is 5. 10 times 10 divided by 5 equals an estimated population size of 20 fish. Here's another example. What is the population size of rabbits in a forest if 12 rabbits are captured in the first set of traps, 3 marked rabbits are captured in the second set of traps, and 8 unmarked rabbits are captured in the second set of traps? Okay, our number in the first capture was 12, and that means we marked 12 animals total and we released them. Our number in the second capture is 3 plus 8, so that's 11. Our number of marked animals recaptured is 3. So let's plug it in. 12 times 11 divided by 3 equals an estimated population size of about 44 rabbits. What is mark and recapture good for? It's good for moving populations, specifically moving populations of non-dangerous and non-threatened animals. If it's dangerous, we don't want to capture them and mark them. If it's threatened, we don't want to capture them because it could hurt them accidentally. What is mark and recapture bad for? It's bad for dangerous animals like polar bears that will attack. It's dangerous for animals that might be harmed by human interaction, also polar bears. It's bad for animals living in clumped populations like migratory birds. It's bad for populations with high degrees of immigration and emigration like migratory birds. And these jellyfish, we can't attach a tag to these jellyfish. There's no way for us to attach something that will stick and will not kill the jellyfish. So this one would fall into number 2, animals that might be harmed by human interaction. And our final sampling type is called number 4, indirect sampling. Here's the method. Number 1, we count specific animal traces in an area, like their prints or their scat, that means poop, or their nests or their burrows, any traces that they leave. And number 2, we use that to calculate population size and population density. This will be an extreme estimation. What is this good for? This is good for moving animals that are hard to find, like mountain lions. They're pretty rare. This is good for animals who might be harmful to humans, again, mountain lions. And this is good for animals who might be harmed by human interaction, something that's so delicate that we can't touch it, or something that's really sensitive to humans and might be harmed by the interaction or the tag from mark and recapture. It's bad for determining accurate population size or population density. It's really only good for an estimation. So those are our 4 sampling types. Our final section is how to sample well. How do we do this reliably and well? Number 1, we need to make sure that our sample is representative of the whole population. Here are some examples of when it wouldn't be. What if we use data from one nursing home to infer the percentage of the US population already infected with COVID-19? This would be a bad sample because the people in the nursing home are going to be a very particular age, and COVID's also going to be spreading through that population very differently than it will through the entire US. We need a sample that's more representative of the ages and the spread pattern of the whole US. No good. What if we only survey bats that are close to a research station for signs of white nose disease in order to determine what percentage of bats are affected? Well, this is a problem because of our location choice. Bats actually avoid light, and sick bats are the only ones that will not be avoiding light, like the lights at the research station. So when we look at our sample, we're going to be seeing a higher percentage of infected bats than we would if we looked over a larger survey area that was not just close to the research station. No good, not representative. What if we only count trees at the edge of the forest to determine species richness? Well, the edge of the forest has different conditions than the interior of the forest. So this is not representative of the whole population and of the whole ecosystem. This would not give us a representative sample. Number 2, our samples must be truly random. In quadrat sampling, we need to make sure that our quadrats are randomly placed over the whole area, not clumped in one corner or just around the edges. We can do this by throwing our quadrat while spinning around so it lands in a random area, or by dividing up our survey area into a grid and using a computer program to randomly choose different locations to put our quadrats. In transect sampling, we need to make sure that the lines are evenly placed or randomly placed over the whole area. They should all be parallel, and as long as they're either evenly placed or randomly placed, that will be a representative sample. In mark and recapture sampling, we just need to make sure that a true random sample of animals is caught. Detail 3, the larger the sample, the better. If we're just using hibernation data from 25 bears to infer hibernating habits of all 200,000 bears in the US, that's not a big enough sample. We could have some errors in there. A larger sample will give us a more accurate estimation. If we're using data from one nursing home to infer the percentage of the US population already infected with COVID-19, again, too small. We need a much larger sample that's also more representative of the whole. The problem with larger sample size is that they get expensive. So in real ecology, sampling is usually limited by budget constraints. And finally, sampling should not disturb the population. In mark and recapture, we need to choose our tags really carefully so that they don't disturb the actual animal. Like, what if this tag changed the behavior of the butterflies and so it made it more likely to be caught later? That's going to change our sample. We also just need to be aware that animals may learn behaviors to help them avoid traps. So choose your traps very carefully, choose your placement of traps very carefully so that it doesn't influence our results. And with that, that's your lesson on population sampling. Go back and catch any details that you missed the first time. Make sure your notes are super complete, turn them in, and we'll see you in class later.